in trying to find the link to the meeting. So first of all, welcome everybody to this EAOG and TSOP joint seminar. So EAOG is the European Association of Organic Geochemists, and TSOP is the Society of Organic Petrology. And we aim to organize a seminar, in fact, every quarter. In practice, it will be 3 times a year, given we have holiday season as well. So my name is Johan Weijers. I'm an organic geochemist, R&D at Shell, but I'm also a board member of the EAOG. And this edition was we organize alternating with TSOP. So this one was organized by EAOG. And for the first time in collaboration with the EAGE, which is the European Association of Geoscientists and Engineers, we were able to try out their WebEx environment, so to make it a little bit more professional. And as you can see, all entered here, it looks very similar to Teams, and all the functionalities are pretty similar. So you can raise your hand if you have a question. You can type your questions into the chat, etc. So that's for the question part. Felix already indicated that if you have a question for clarification, please feel free to just ask it during the lecture. If it's more a real discussion point, then maybe better to save it to the end. Normally in these joint seminars, we have Joe Curiali as a moderator, but unfortunately he couldn't attend today, so I'm afraid you have to do with me today. I'll try to do my best replacing him. Also in agreement with the speaker, we record this session. So for those members who cannot attend, there is still the opportunity to look back the lecture of Felix. And speaking of members, if you happen not to be a member yet of either TSOP or EAOG, please consider becoming a member. It's a small effort, but to us it means a lot. It definitely helps us building a community. So please reach out to the respective websites of us or to us personally. So with that introduction, now let's move on to the introduction of the speaker, which is Felix Elling. And then I quickly move to my notes. So Felix studied marine geosciences in Bremen and obtained his MSc degree in 2012. And then continued with a PhD at the same institute, but at Marum, where he obtained his PhD cum laude in 2015, based on his thesis, Factors Controlling the Lipid Composition in Marine Planktonic Thalarchaeota. And after a short postdoc, also in Bremen, Felix moved to Harvard for a postdoc with Professor Ann Pearson. And in 2021, he came back to Germany, and since 2021, he's the group leader of the Molecular Geobiology Group at the Christian Albrecht University in Kiel. And here at Kiel, he and his group study molecular fingerprints of present and past microbial life and their interactions of microbes with geochemical cycles and climate. Very nice to note that Felix is actually the recipient of the 2021 Peter Schenk Award, which is an early career award from the EAOG for outstanding contributions to organic geochemistry. And the presentation that Felix will give today is in fact an extended version of the presentation that he gave at the IMOG. This is the International Meeting on Organic Geochemistry that was held in Porto a few weeks ago. And his abstract was selected by the scientific committee as basically the best abstract of all that were submitted. And therefore it was awarded with the Geoff Eglinton Award, which recognizes innovative research in organic geochemistry that explores novel ideas and techniques in science. So therefore we are very glad that Felix is willing to provide this lecture again here in this forum. On this fascinating story on quinones. And I leave the introduction of that topic to you, Felix. So over to you.
Thank you, Johan, for the introduction, and thanks for the invitation to speak to you today. So, so I'm presenting research that I started initially as even as a PhD student maybe, and then escalated a bit during my postdoc, but was basically always a side project. And then that's how it started, but then the majority of the work was actually then done in Kiel. And it involved many, many collaborators. So it's about 20 collaborators that are listed here, so I cannot thank them all here, but I'll let some of the collaborators throughout the talk. And so today's talk will be about quinones and their biosynthesis, and specifically about a group of bacteria that are in the phylum Nitrospirota. And so the quinones that these Nitrospirota produce can tell us something about the evolution of aerobic metabolism. So what are quinones? So quinones are redox-active isoprenoid lipids. So they have very diverse structures. So what you can see for each of the different types of quinones, they all have a cyclic moiety, and they have moieties that can be reduced and oxidized. So these quinone groups here, so they occur either in this form where they have just one hexyl ring, so this benzoquinone, or with two hexyl rings, and this is called a naphthoquinone. And as you can see, organisms like to decorate these molecules, so they have additional functional groups added to these rings, and those differentiate the structures in addition to the isoprenoid chain that is attached. So the isoprenoid chain is relatively long. It's usually between 4 and 12 isoprenoid units, so basically C20 to C60. And they can have varying amounts of unsaturations in it. So if you combine all of these structural types and the isoprenoid chain length diversity, so you will end up with quite a huge number of possible structures. And so these compounds are produced by basically all life forms. There's very, very, very few organisms that do not produce any of these. And so the reason why every life form needs it is because they're essential for electron transport. So basically any energy metabolism of a cell is based on transporting electrons. And this electron transport happens in the membrane. And a crucial part of that is mediated by quinones. So what quinones do is they can transport electrons between different enzymes or proteins in the membrane. Because they are lipids, they are soluble in the membrane, and they can actually travel through it. This is important for respiratory complexes that are used to breathe oxygen or other substrates. Can also be anaerobic respiration. And the reason why quinones can do this is because they can be oxidized and reduced. So on the left side, we see the oxidized quinone. So this is a quinone called a ubiquinone. And when you add 2 electrons to it and 2 protons, then you will get a reduced quinone, which you can see here. So, and in this, what the enzymes in the electron transport chain do is actually they receive a quinone and then they either reduce or oxidize it. And then they can, in this way, transmit or take up electrons. All right, so then this flow of electrons is important for each cell, and that's the reason why every organism has developed to use some form of these quinones. And now this is kind of like a textbook explanation. This process has been known since about the 1970s or so. So it's, I'm telling you nothing new here. This will be found in any biology textbook. But the reason, so to go back a little bit historically, how I came to deal with these molecules is basically by accident. So I'm an organic geochemist, right? So I'm used to study organic molecules, lipids. And when I started my PhD, I had no idea what quinones were. I had not ever heard of the term even. Maybe if I had read a basic biology textbook, I would have, but apparently I didn't. So anyway, so what I did during my PhD, I studied the lipidome of archaea and specifically of Saumarchiota. And so this was back in about 2012 or so. So that were the early days of high resolution mass spectrometry applied or coupled to liquid chromatography. And so in here we can see so-called density map. So it's basically a heat map of ion occurrences. So retention time is on the x-axis, and then on y we have M to Z ratios. And so basically all of the green compounds are, well, every colored feature, so every blue dot is basically one lipid, one compound, and all of the green features are the features that we could easily identify in these archaea. And so this took quite some time. So I spent many hours in my PhD just staring at mass spectra, and especially at those density maps. And something that always popped up were these compounds in this lower to middle mass range, so about 400 to 600 or so. And there were plenty of those peaks. I could never identify them. So in our group at Maum, we had always gave them, had always given them some preliminary names that didn't really make sense. For example, we called them a wholly unsaturated bifurtane or something because the mass would fit, but still we didn't really know what it was. And so then one night, when I was working very late on these spectra, and I read some completely unrelated literature of microbiology of some organisms, I came across one of these masses that I always saw. And I thought, wow, okay, now I finally know what it is. It is a quinone. And so this was some literature from the 1980s or so. So people apparently knew about this very early. And then what I did is, it was a bit too late to go back to the mass spectra and try to figure this out. So I just sent an email to my fellow PhD student who was also studying lipidomes at this time. So his name is Kevin Becker. And what he did, so I was always coming in very late to work and staying late, and he was always coming in very early. So basically I sent him an email, and then by the next morning, he already gave me an answer about all the mass spectra. So he had already looked through all the archaeal lipidomes that we had and checked for the presence of quinones. And so basically what he found is that the peaks that I identified was actually contamination. But there were the other peaks next to it that were the actual quinones. And so this kind of started us on this kind of unexpected journey, because for neither of us, this was the topic of our PhD. So Kevin's topic was about lipidomes, or environmental lipidomes, and mine was about lipidomes of Saumarchiota, but not about studying what quinones do and how they are distributed in archaea. But basically, the next year of our PhD, we spent doing only this. So we looked at all the different archaea and the quinones, so we could identify a lot of different quinones. So here's a representative mass spectrum. And so this is for a so-called, so this is mass spectrum for so-called menaquinone 6. It's actually the simplest mass spectrum you can ever look at. So it basically has a base peak molecular ion, and then has a single superdominant fragment ion. So once you know what you're looking for, it's actually super simple to identify. And as I said before, this had already been identified before in Archaebacteria, but yeah, that was in the 80s, and to my shame, I had not read that literature before. All right, so what we then did is there's the figure that I wanted to see. So this is the quinone that we identified, which we could then show that this is highly specific for Saumarchiota, and that we can then use to trace those organisms in the environment. All right. Then we obviously discovered that we had missed out on the whole diversity of quinones that we can find in basically all the samples that we ever analyzed, because our method of choice could resolve those very well. And so then we went back to our old data from the Black Sea. So this is showing water column profiles from the Black Sea. On the left, there's a very simplified schematic of the different redox zones of the Black Sea. So at the top, we have a photic-oxic zone. Below that, we have a chemocline that occurs in the depths, depending on where you're on the Black Sea, at around 80 or 100 meters. there's a photic anoxic zone below it, where green sulfur bacteria live, and then basically from around 200 meters or so to the bottom, in this case it's 1200 meters, there's completely anoxic dark sulfidic waters. So the Black Sea has a very, very strong and also a temporarily very stable redox stratification. And so because we knew that the quinones were related to redox processes, we thought, okay, well, then they should be able to resolve the stratification of the Black Sea and tell us something about who lives where. And so if we go from left to right, we can go through the different zones of the water column. So shown here in green is vitamin K1. This is a quinone that occurs exclusively in photosynthetic organisms, specifically oxygenic photosynthesizers. You can see it's only present in the photic anoxic zone and then basically absent in the rest of the water column, just how we expect. For ubiquinone, this is a very general quinone produced by aerobic metabolisms. We could also see that this is mostly present in the oxic water column and then kind of diminishes in the anoxic water column. There are some really weird isomers of this that we only find in anoxic regions, but this is a topic for another day. The quinones that we identified in archaea are also peaking exactly where we expect our aerobic archaea to live. And we can find in yellow the quinones from the green sulfur bacteria, which basically inhabit a very, very narrow zone of the water column. And then in the deep anoxic zone, we do see a diversity of quinones that are associated with anaerobic metabolisms. For example, this from methane oxidation, this MQ called compounds, the MK, which is menaquinone, so it's the type of naphthoquinone that occurs basically exclusively in the anoxic part, and then quinones associated with methanogenesis. So this kind of took up a lot of our time going through all of these data, and it was very exciting for us because we thought, okay, we can use this as a very nice biomarker. But then this wrapped up around 2015. I went on to my postdoc and kind of never really found an application for these quinones anymore. Was also distracted by more exciting topics at that time. And specifically, I got interested in hopanoids. I went to study hopanoids in nitrate oxidizing bacteria. So they are very ubiquitous in all environments, soil, ocean, lakes, and so on. And I did this together with Thomas Evans, who's now at Shell and was back then a postdoc of Roger Summons. And so we are just interested in finding, was basically a side project of my actual postdoc project to just see what interesting hopanoids they produce, because I thought there might be some prolific producers in marine environments. And so as part of this side project, we developed another side project because I thought, okay, for the fun of it, let's just look at what quinones they make because nobody had ever looked at that. And so to my surprise, having looked at hundreds of cultures for their quinone content, I always found the same quinones that were already known. But in this organism, there was an unknown quinone. And it didn't take us very long to decipher what it is, because the spectrum, the the fragmentation spectrum, so the MS2 spectrum was very easy to interpret. So we call this a mesylplastoquinone. It is composed of this benzoquinone moiety, and it has 3 extra methyl groups attached to it, and then a standard isoprenoid chain. So at this point, we only have this mass spectrometric confirmation, but later on, we also got NMR data on this, but I'm not going to show these today. All right, so then at this point, so to use this might not look very exceptional, kind of boring looking, okay, well, it has some methyl groups here and there. Why do we care about this? We do care about this because the functional groups that are attached to the ring moiety alter the redox potential of the whole quinone. And if the redox potential is altered, this means that the type of metabolism that can be done with it, well, the type of metabolism that can be done with a specific quinone depends on its redox potential. So what we did then is to look at more diverse organisms from the same branch of bacteria. So this branch of bacteria is called the Nitrospirota. The Nitrospirota are named after the first discovered organisms from this phylum, which are the Nitrospira. So it's the class Nitrospiria, and the genus is Nitrospira. And they are aerobic nitrate oxidizers. Then in this phylum, there are other classes that have been cultured, for example, the Leptospirilia, which are aerobic iron oxidizers. The Meganitropus, which are aerobic manganese oxidizers. And then there's only one branch of this phylum, this TSOP, which are anaerobic sulfate reducers. So by and large, this is a phylum of very diverse aerobic organisms that all do redox cycling of some critical elements. So either it's nitrogen, iron, or manganese. And so when we looked into these cultures, so we cultivated them and extracted their quinones, and we saw that all of these aerobic strains made this new compound, so the mesoplastoquinone, which is abbreviated here as MPQ. You can see that it kind of differs. So the nomenclature here is the first number gives the number of isopenoid units, and then the second number gives the number of unsaturation. So this mesoplastoquinone 7,6 has a 7-member, or so 7 times 5-membered isopenoid chain, so C35, and it has 6 unsaturations in the chain. And then only the anaerobic member of this phylum, the Thermodicylprovibrio, contains menaquinones, which are typical for anaerobic metabolisms. So what makes this even more interesting then for us is the wide distribution of this, right? It's not just a single organism, it's basically a whole phylum of bacteria that makes this. And it's super relevant because the structure is very similar to quinones that we already know from other organisms. So as I said, the mesoplastoquinone has 3 methyl groups attached to the ring. The most closely related structure comes from cyanobacteria. It's the so-called plastoquinone. It's identical to the mesoplastoquinone, except for this missing methyl group here at the C2 position. And this is quite relevant because the plastoquinone is involved in photosynthesis, and it is the only use for this compound is for photosynthesis. So obviously we know that Nitrospira does not do photosynthesis because it grows in the dark and only in the dark. But we did think, okay, this needs to have some relation to aerobic metabolism. If we look at the other very well-known quinone that is involved in aerobic metabolism, this is also a benzoquinone called ubiquinone, and it shares structural features with the plastoquinone and the mesoplastoquinone. So we have this common benzoquinone moiety, but then on the C2 we have a methyl group, same as is preserved in the mesoplastoquinone, but not present in the plastoquinone. And then on the C5 and C6 position, there is methoxy groups, which are absent in the other 2 molecules. So from what this is showing is that the mesoplastoquinone has features that are preserved in the other 2 types of quinones that are related to aerobic metabolism, but still has, so it combines those features in a distinct way that is not found anywhere else in all of life. All right, so what makes those compounds interesting is in regards to energy metabolism is that, as I said earlier, the structure of the compound is related to the redox potential of the compound. So as I said earlier, the anaerobic organisms mostly use this so-called MK menaquinone, which has a very low redox potential. And so if an organism would want to do aerobic metabolism with this menaquinone, this can be quite challenging for the organism. It has to do with the redox potential of the menaquinone. So the redox potential of the menaquinone is around minus 100 or so. So you can see the menaquinone data point in this graph here, which shows redox potential from negative to positive. This is black diamond. And the if it were to oxidize oxygen, then it would need to transport electrons between two enzymes, or two parts of enzymes. So one is an iron sulfur cluster, and the other part is a so-called heme. We don't need to concern ourselves with the details of that, but the point is that it needs to transfer. The point of this is that the quinone always needs to have an intermediate redox potential in between those two enzymatic features. And if the redox potential is negative, that means that it comes into range where the quinone itself can react with oxygen. And if it reacts with oxygen, then it donates a single electron to the oxygen, which then forms an oxygen radical, and that oxygen radical is super damaging to the cell. The way that life has found a way around this is to increase the redox potential of both the enzymatic components and of the quinones, so that the redox potential of the quinones is in the positive range. And so here in blue, I'm showing the ubiquinone, and then in green, sorry, the structures are missing, I'm showing the plastoquinone. And we call this variant of electron transport a high potential electron transport because it has a high redox potential. And by doing that, the quinones are outside of the range where they can spontaneously transfer electrons to oxygen. So basically, if you're an organism performing any type of metabolism with oxygen, that could be photosynthesis, or it could be just aerobic respiration, so just breathing oxygen, then in this case, you would want to have such a high potential electron transport chain, and for that you need a high potential quinone. And so the interesting part about those high potential quinones is that they have a very limited distribution. So this is shown here by the colored branches. We're looking at a tree of life. It shows 3 domains of life: bacteria, archaea, and eukaryotes. All the branches that are in black use MK for anaerobic respiration, so menaquinone. The branches that are in blue use ubiquinone for aerobic respiration, and the branches in green use plastoquinone for oxygenic photosynthesis. So it's this branch here, the cyanobacteria, and then the plants. And so the reason why eukaryotes can do this in the first place is because eukaryotes have inherited 2 features from bacteria. They have inherited mitochondria, and they have inherited chloroplasts. And those 2 brought their quinones with them. So the mitochondrial quinone is ubiquinone. So we as humans, for example, all animals, all eukaryotes have mitochondria, and in our mitochondria we do have ubiquinones. And all oxygenic photosynthesizers, so plants, algae, cyanobacteria, inherited the plastoquinone from the initial chloroplast that was transferred. So we have a very strict separation of these, and this can basically kind of this world where we either have this anaerobic quinone or we have 2 options for aerobic quinones. But now with the mesoplastoquinone that we found, suddenly we have a 3rd option. There's a 3rd high potential quinone. And so this is why we got so excited about this molecule, because it is not an interesting new structure. It's not only occurring in an interesting group of bacteria, but it is also related to the evolution of aerobic metabolism. All right, and so if we wanted to put the evolution of aerobic metabolism on a time scale, so this is a highly stylized version of this. So we see this time arrow here that is going over the 4 billion years before the present to about 2 billion years before the present. And well, this is something people can argue about for hours, but we could say that bacteria evolved sometime between the formation of Earth and about 3.5 or so billion years before present. And at that point, we know that there was definitely no oxygen in the atmosphere. And so bacteria initially probably used this quinone, menaquinone, for their anaerobic metabolism. Then we know that at some point, there was oxygenation. So around 2.5 billion years before present, oxygenation in the atmosphere started. We know that after this time, we have widespread aerobic metabolism. There's all kinds of aerobic metabolism, such as iron oxidation happening. We know that photosynthesis was, so oxygenic photosynthesis was producing that oxygen. And so at this point, the ubiquinone and that is then used for aerobic respiration must have evolved. But we don't really know anything what happened in between. So, for example, did the plastoquinone that is needed to produce the oxygen, did this evolve during the oxygenation, before the oxygenation, or at some other point? And so the problem with this is we cannot check this in the geologic record. There's no fossil record of quinones or other electron transporters that would go back this far. We can address this from a different angle, and that is from the genetic perspective. And so to do this, we first need to find out how or what are the genes that are responsible for producing the different high potential quinones. For the ubiquinone, it is well known how it's synthesized in bacteria. It has a 9-step pathway, so there's some variations to it, but in principle, all ubiquinone producing bacteria and also eukaryotes use this pathway. We don't necessarily need to go into all of these steps, but just to give you some overview, this biosynthesis pathway starts with chorismate. So this is coming from the so-called shikimate pathway, and it's also the pathway that provides the aromatic precursor for aromatic amino acids. And so this branches off from the aromatic amino acid biosynthesis. The pyruvate here from chorismate is removed. We end up with this so-called 4-HBA that is then prenylated with prenyl chain, and then we have structural modifications that add, for example, the methyl groups here, add hydroxylations, and add methoxyl groups. We don't need to understand this in detail, but the key takeaway here is that we do have very well-described high potential quinone biosynthetic pathways in bacteria. And so we can use this knowledge then to probe in our Nitrospirota organisms whether they use a similar pathway. And so because at this point we did not know anything about how they are biosynthesized in Nitrospirota, we used labeling experiments. So first of all, so I'm showing 2 experiments. One is with 13C labeling. And so we were interested in finding out if they use the same intermediate, so the 4-HBA. Because this is a central intermediate that gets prenylated, we should, if we feed this 4-HBA in a isotopically labeled form, then we should see this incorporated in the quinone that we can analyze using HPLC-MS. And so we took 4-HBA that has all the 6 ring positions labeled with 13C, and then checked either control cultures that were fed with non-labeled 4-HBA or the cultures that were fed with the isotope labeled 4-HBA. And so you can see in the MS1 mass over here that we have an increase from 780 M to Z to 786. So there's a 6th increase, which indicates label incorporation. And when we look at the MS2 spectrum, that we see that the characteristic fragment, which is this benzoquinol moiety, its mass increases by 6 units from 165 to 171 mass units. So we do get a full incorporation of the ring into the molecule, which means then that 4-HBA is the precursor as seen in other bacteria. And then the other distinct feature of this quinone are the methyl groups. We know from other bacteria that in the ubiquinone biosynthesis, the methyl group is at C2 is added by one specific enzyme that uses methionine. And so methionine is an amino acid that contains a single methyl group that's connected to the sulfur. And so this is easily cleaved and can be transferred to a new molecule. And there are different ways of doing this. I can't go into detail about this. but we specifically fed it with deuterium labeled methionine. So this has deuterium, so 2H at all three, all three hydrogens of this methionine are deuterated. And so what we saw when we fed this to the Nitrospirota was an incorporation of these three deuteriums. But because methylplastic quinine contains three methyl groups, we saw incorporation of 3 times 3 mass increase, so a mass increase of 9. So the mass increased from 780 to 789. So which means that this methyl group gets fully and intactly incorporated into the molecule. And so because the different methylating enzymes either fully transfer these deuteriums or they transfer only part of the deuteriums, we know that this enzyme must be functionally very similar to the one that we find in other bacteria. So basically, if they incorporate all 3 of the deuteriums at each position, we know that it must be identical. If they are less than those, so if we see, for example, incorporation of only 6 or 3, then we know that it is a different enzyme. All right. So this way, we already knew that we can use a bacterial pathway, but the ideal way is, of course, to do this genetically. So you knock out the genes and then you identify if the gene of interest is involved in the pathway. So we had some hints that we could do this because we looked into the genomes of Nitrospirota. Here I show 3 genomes of the genus Leptospirillum. And we could identify that there are some gene homologs, which are here in different colors, that occur in all the genomes and that also have some similarity to the genes that we find in other bacteria. So, for example, one is a quinyltransferase and the other one is a methyltransferase. And so with this knowledge, I got into contact with 2 collaborators, Adrien Pierrel and Sophie Abi, who are at the University of Grenoble. And they are expert in the quinone biosynthesis pathway in E. coli. Obviously, from the start, we knew that we could not do such experiments in Nitrospirota. And the reason for this is that the quinone biosynthesis is a very essential element of the cell. And so if we knock this pathway out by deleting a gene, for example, we will just kill the organism because it cannot produce quinones and then it cannot reproduce. And so our idea was to implement the genes from Nitrospirota instead into E. coli. E. coli is obviously the workhorse of molecular biology, so it's easy to transfer genes into it, but also it has a big advantage that it has, as one of very few organisms, it has 2 quinone biosynthetic pathways. And so if you knock out one of them, it hurts the organism, but it doesn't kill it. And so what we did is we engineered E. coli to make a very basic quinone that misses all of the functional groups. For example, when we knock out 3 genes, then we can miss all of these extra functional groups at C2, C5, and C6. Then we add the gene from Nitrospirota in a plasmid and express it in E. coli, and then we end up with E. coli that then produces specific modifications to the quinone that then are related to the gene of interest that we introduced. And so we did this for dozens of genes from Nitrospirota, so I will spare you the details on that. But in the end, we could implement the full pathway from Nitrospirota into E. coli, and we got E. coli to make methylplastic quinones. So that's showing here. So this is showing that in purple, this is methylplastic quinone produced in milligrams per milligram of cell. And irrespective of which Nitrospirota species, so this is shown for 3 species, we add the genes to E. coli, we can produce the methylplastic quinone. And this was actually quite convenient because E. coli works very well, so we could produce very large amounts of biomass from E. coli, harvest the mesoplastic quinone, and then do NMR analysis on it, which we could not do before because we could not produce the required amount of biomass. All right, so once we know what the pathway of biosynthesis is, we can analyze the evolutionary or the phylogenetics of the genes, so basically the evolutionary relationship. And so this figure, which is a bit complicated and overwhelming, shows the biosynthetic pathway that we characterize. So in blue on the bottom is the ubiquinone biosynthetic pathway. And on top is in purple the mesoplastic quinone pathway, and in green the plastoquinone pathway. So the one that is found in plant and bacteria, cyanobacteria. So, and as you can see, this is shown by the shared colors and also by the connectors here, connecting lines. There are many pathways that are biochemically very similar. So basically all these steps, except for those that are specific, that produce a specific structural variation of these molecules, are identical biochemically. And we always find genes which are given by these name or these abbreviations that could be, or that we identified as responsible for this step. And so then we ask ourselves, so if those pathways are obviously biochemically very similar or almost identical, then they must be related to each other, but how are they related? So which pathway evolved first, and where did it come from in the first place? And so for this, we zoomed out a lot. So this is showing a tree of over 500 bacterial genomes. So this covers all of bacterial diversity as we know it today. And then we analyzed the occurrence of the specific genes for the different high potential quinones. And these are highlighted by the colors. So again, it's the same colors as in the other figures. The mesoplastic quinone is shown in purple here on this outer ring, the plastoquinone in green, and then the ubiquinone in blue. So from this, we can see that the distribution of these genes is very limited. It's limited to these three phyla, so to the Pseudomonadota, which are the producers of the ubiquinone. The mesoplastic quinone is limited only to the aerobic part of the Nitrospirota, and then the plastoquinone is limited only to the cyanobacteriota. And in the all other organisms, there are only melaquinones or other quinones being present that are not high potential. So if we expect those pathways to be directly related by vertical descent, so basically from their parent to daughter, just over trillions of generations, then all of these should cluster together. So in this species tree, they should all be branching out next to each other. But as you can easily see, they don't. So there are these gaps in between. And so our high potential quinones only form these kind of islands in the sea of other bacteria. So this means that the genes cannot have been transferred in a vertical fashion. All right, so then we looked not only at this top-down view from bacterial phylogeny, but we looked at the phylogeny of the individual genes. And so here I'm showing the phylogeny of two genes. So from this, we can show that the genes are related to each other. So the color coding is again the same. So we have the mesoplastic quinone in purple, ubiquinone in blue, plastoquinone in green, and then the melaquinone in black. And so for the two steps that I picked, so the quinone transferase and the decarboxylase, we can see that the order of this branching pattern is always the same. So all the genes for the plastoquinone plot together, or the ubiquinone plot together, and they plot together in the same relationship with regards to each other. So this tells us that there is neither of these reproducers of high potential quinones were the first to produce it. So if you go back this genealogical tree, you can go back all the way to this earliest branching point here, where there's branching between the melaquinone and the high potential quinones. So this tells us that by the time that the last the earliest organism produced the menaquinone, so the anaerobic quinone, there was already an organism that had some version, some maybe very ancestral version of this high potential quinone biosynthetic pathway. But what we can also see from this branching pattern is that this organism does not exist anymore. So it was an organism that died out some point before the present. So it could have been billions of years ago. It could also be have died out more recently, maybe during so I lost my thread here. So it could have also died out more recently. Or it could be an organism that we have not simply sampled yet. It could be still out there. So, but from the genomic perspectives that we have today, it must have been an event that happened very, very early in Earth's history. And so what we can do is we can actually plot this on a time axis, because we know one crucial thing, and that is that the last common ancestor of each of cyanobacteria, Pseudomonas adotta, and the Nitrospirota was producing the respective quinone. And so if we know the age of each of these phyla, then we know the age of those quinones. And so we can come up with some kind of hypothetical evolution of quinones. So there was a last common ancestor, the last universal common ancestor, about 3 billion or 3.5 billion years ago. There's a branching in the evolution that goes towards anaerobic organism and a branch that goes towards developing aerobic metabolism from some probably ancestral quinone that we cannot reconstruct fully. But we do know from the structural features and the genes that it probably already could have methylate at the C2 position. And then later we have this branching that we cannot place in evolution very well into these 3 branches that are then transferred to the plastids, so the chloroplasts and then the mitochondria, with some of these specific adaptations, such as the methoxy groups or the extra methyl groups, being added later. And so now to put this on a time, so this is just like relatively speaking, but to put this on absolute time, we have to go to another somewhat complicated figure. So this is showing, again, the time between 4 billion years and about 2 billion years before the present. And there are all kinds of events on here. So the colored bars show the age estimate of each group that is shown. For example, the crown bacteria, which means like the last common ancestor of bacteria, somewhat around 3.5 billion years at the latest. The cyanobacteria can have a very large range, but if we focus only on the oxygenic ones, then their range dates back to about 3.2 billion years before the present, and then the Pseudomonas adotta as well to the same time. The aerobic Nitrospirota, we do not have enough data, so we cannot put them on the timeline. And then the latest occurrence of these clades must have been around the time of the so-called GOE, which is the Great Oxygenation Event. So the first time in Earth's history when atmospheric O2 levels rose, or they rose to a high level and also stayed at that level. And so if we now take these age estimates, then we know that the high potential quinones must have been around by the time before each of these big groups evolved. And so that places the evolution of quinones around 3.2 or to 3.4 billion years before the present. And so what you can easily gather from this is about 800, 900 million years or so before there was appreciable oxygen in the atmosphere. So then what our reconstructions show is that there was the high potential quinone already present in diverse bacteria way before there was oxygen available to breathe. This seems a bit counterintuitive at first, but if we look at other evidence, there's actually quite a bit of evidence that there could have been aerobic metabolism, so something using or producing oxygen. For example, there's geochemical evidence for localized oxygen presence, so-called O2 oasis around 3 billion years. There is an expansion in the diversity of genes called, or enzymes called O2 reductases that handle oxygen. There's radical oxygen species protection mechanisms evolving during this time as well, and other evidence that there's an overall burst of diversity around the same time. So what we cannot definitely prove that. What we can prove is that there were organisms that were capable of breathing oxygen during that time, but we cannot necessarily prove that they were actually breathing oxygen during that time. All right. So with this, I want to conclude. Put up this somewhat simplified timeline, showing again the same time frame, but now we would put the evolution of the primordial, so the ancestral aerobic quinones and aerobic metabolism, at least sometime before there was appreciable oxygen in the atmosphere. And so with this, I would like to thank you for your attention, and I'm very happy to answer any questions.
Thanks a lot, Felix, for a wonderful presentation. So yeah, I open the floor for questions. Please feel free to come off mute if you have one. And while you might think of a question, I might start off, Felix, with one small one. You showed at some point the mass spectra of those quinones that you had deuterated. It was interesting to see that the mass fragment, a very typical mass fragment of those quinones, was suppressed actually in the deuterium labeled species. Is that something that you see more often?
Yeah, so we have since used this a lot. I can just go back to that figure. Yeah, so this kind of works very well in LCMS because to get these compounds, for example, on the GCRMS to measure the isotopic composition would be a whole can of worms of method development. So the good thing is we do feed quite a bit of this label so that there's a high chance of getting the label into the cell. And it's also metabolites that easily enter the cell. And so what you then get in your MS1 spectrum is actually you get all the several peaks. You will get the peak without incorporation, and then depending on how much label you added, you will get one with 3 extra mass units, and then 6, and then 9, and so on. So I'm showing only here the +9 labeled one. And with the LCMS, so especially with the high resolution LCMS, and if you have a control that is unlabeled, you can actually relatively easily calculate the incorporation rate of your label. So it's basically you're doing isotope ratio mass spectrometry, but with a very high error. So
yeah. Thanks for that. Any other questions online? Not yet, then I'll ask just another one, if I may. You said there's no fossil evidence of those compounds, the quinones. Is that because there is just not found a suitable fossil record for it, or is that because we do not know the fossil representatives of those molecules?
I think it's probably both. I want to preface this by saying that I think nobody has really looked for them, either for the compounds themselves nor their degradation products. And so I think that is a completely open book, and certainly exciting to look into. I wouldn't expect those to survive like 3 billion years, probably unreasonable.
Yeah.
I know like several tens of thousands of years is no issue detecting them. We have not looked further. So it's definitely something that one should look into. If they survive like hundreds of thousands of years, maybe millions of years, I could imagine in a good preservation environment. Probably even more than that. So because something like tocopherol, for example, can be found in some even Cretaceous sediments during OAEs. So a quinone is structurally very similar to that. So I could imagine that surviving.
Yes.
Yeah.
We do find tocopherol as vitamin E sometimes in oil or extracts.
So what age are those?
Oof, that's a good question. I have to go back to that particular one, but probably low mature material for sure. But on the other hand, we could of course also think about what could a degradation product look like if you take out those two oxygen atoms from the molecule. How would it look like? Because what we do often see in, for example, source rock extracts are those phenyl benzenes, phenyl toluenes, and phenyl xylene, if you wish. So exactly with these different types of methylation patterns. But are those related? That's in the question. But maybe something interesting to look into.
It's definitely very interesting to look into. So I would say, yeah, so maybe the oxygens are the least stable part of the molecule. Everything, and the double bonds, of course. Everything else, I think, would have some chance of being preserved. And then it's a question like how well the side chain survives degradation. The side chains are often relatively long, so 8 isophenol units is typical. But there are plenty. So, for example, vitamin K1 that's also used in photosynthesis has, for example, a phytonal chain. So that would be very fitting with a phytonal, yeah, or some kind of benzene phytonal combination.
Yeah, that would be an interesting topic indeed to further explore. All right, we're hitting the hour. Are there any more questions online or in the chat, perhaps? See, the chat is empty. I think it was a very clear story then, Felix.
Yeah, thank you so much.
So if there are no more questions, and since we are just over the hour, I propose that we close the presentation here. So big thank you to Felix once again. Thank you all for dialing in. And we hope to see you again during the next joint seminar from EAOG and TSOP. And then I wish you a good rest of the day or start of the day for wherever you are on this globe. Thank you very much, and hope to see you later.
Thank you. See you later.